Hello again, students. We've finally made it to essentially the last step of our central dogma, which is translation. In this lesson, we're going to look at how that mRNA that we just made in transcription can be converted into proteins in the process of translation. So remember that in transcription, we can make 3 types of RNA. Now, all of these are going to be used in translation. Our mRNA is going to be the blueprint. The rRNA will make up ribosomes, which are going to hold everything together in translation. And our tRNA will bring our amino acids to help form a protein. So we can think of our rRNA and tRNA like the machines that are working on converting that mRNA into a protein. As I mentioned, translation is going to be the process of converting RNA into proteins. So we talked before how we went from DNA to RNA in the process of transcription. Now we're taking that RNA and turning it into a protein through the process of translation. And we can see that this is occurring in the cytoplasm. So this process will be in our cytoplasm. Now, to help you guys remember which comes first between transcription and translation, we can think translation happens later. Translation happens later. So we can see translation here is our second step. Essentially what we're doing is we're taking that mRNA that we just made in transcription, we're bringing in amino acids, and we're forming a polypeptide, which will make up proteins. Now before we jump into the steps of translation, I want you guys to be familiar with a couple words here. Our first is a codon. This is going to be a 3-nucleotide sequence that will code for an amino acid. And this is going to be part of our mRNA. So if we look down here, we see our mRNA strand. And we can see these 3-letter sequences here. So we'll use the example of AUG. And this is actually the start codon for translation. Another important term here is an anticodon. So this will also be a 3-nucleotide sequence that's going to be complementary to each codon. But in this case, this will be carried by our tRNA, or our transfer RNA, which we can see over here. So every tRNA molecule is going to carry an anticodon, which will match up with the codons of mRNA. And it'll also carry an amino acid. In this case, we have lysine here, so this is an amino acid. Every tRNA molecule will bring in an amino acid and an anticodon. Now let's look at the actual process of translation. In our first step, we can see that our mRNA template here is clamped in our ribosome. So this is our ribosome. And remember, this is made of RNA combined with proteins. So this mRNA codon is going to attract an anticodon of tRNA. We actually saw that AUG was the start codon. So the start codon brings in the first tRNA molecule and gets the process going. Next, our tRNA molecules are going to keep bringing in these amino acids, and we're going to start forming peptide bonds between those amino acids. So as this keeps going, our tRNAs keep coming in, matching their anticodons to the codons of mRNA. These amino acids will be strung together, and we start to form a polypeptide. In our last step here, our ribosome will reach a stop codon. This polypeptide that we made will be released, and our complex of our rRNA, our ribosome, our tRNA, and mRNA will disassemble. So this polypeptide will be released, and this will go on to fold and become a protein. Now, when we talk about the genetic code, we can think of the words as codons. And remember, those were our 3-letter sequences from our mRNA. Now, we only have 20 amino acids, but there are 64 different codons. So what does this mean? This means that multiple codons can code for the same amino acid. For example, we can see up here that we have 6 different codons that code for the same amino acid, leucine. We can also see that we have one start codon, which we talked about before. This is AUG. So this codes for the amino acid methionine, and this AUG is what gets the process started. We also have 3 stop codons here, so our UAA, UAG, and UGA. These will be found up here. And I want to point out that these stop codons do not code for amino acids. So we can abbreviate amino acids as AA. Our stop codons will not code for any amino acids. We can see that every other codon in our chart here is associated with an amino acid, but our UAA, UAG, and UGA are just stop codons. They're not coding for any amino acid. So let's do some practice here. I'll give you a codon. Let's just say CAG. And I want you to tell me what amino acid this codes for. In order to do this, we're going to look at our first letter, which is C. Our first letter is C. Our second letter is A, so we know we're going to be in this box. And our third letter is G. So we can see the amino acid associated is glutamine. So our CAG will code for the amino acid glutamine. Let's do another example. Let's do GUC. So our G is down here. Our U is right here. And our third letter is C. So we can see that this will code for valine. Now I wanted to connect this topic of translation to something going on in our world right now, which you are all far too familiar with, which is COVID-19. So the interesting thing about the vaccine for COVID-19 is it is an mRNA vaccine. So this essentially gives our cells the instructions to make proteins, right? We just talked about translation where we start with an mRNA and we translate that into proteins. So in the case of the COVID vaccine, our cells will translate that mRNA into proteins, specifically these spike proteins. And these are the proteins that are found on the surface of the coronavirus. So our cells will use the instructions from mRNA to make spike proteins, and they'll put these on their surface. Then these proteins can be recognized by the cells of our immune system, and our bodies will have an immune response. So essentially, our bodies learn that, hey, this protein shouldn't be here. It's an invader. So they'll make antibodies so that we can recognize this spike protein again in the future. Then if the virus enters the body, the antibodies will recognize that spike protein and attack the virus and ultimately prevent us from getting coronavirus. At this point, we've now gone through our central dogma. We've seen how DNA can be copied in DNA replication. We've learned that DNA can be transcribed into RNA, and we just learned how that RNA can be translated into proteins. That's it for this lesson. If you guys found any of this confusing, please rewatch the video and let me know if you have questions.